All right, this is where we left off. Blood typing.
Yes.
We did the ABO just as a review. Remember that in the first diamond, this ABO system is the exception. And that these antibodies, remember antibodies are globulin proteins. And anything that causes your immune system to make globulin proteins is an antigen. And so we have A and B surface antigens. This is the exception because if you have A antigens, your type A blood, and without ever being exposed to B type blood, you are born with anti-B. This is the globulin in your plasma. It's the plasma protein globulin. Same thing with B. If you have B surface antigens on your red blood cells, your type B blood, you have anti-A in your plasma without ever being exposed to the A antigen. That's why it's an exception, because when we think of making antibodies, we are thinking in terms of like the flu shot, where you have to be exposed to have the antibodies. That's the general rule. This is the exception. Everybody understand the exception? So you gotta remember this first diamond, and you gotta remember the last one. No one ever makes antibodies to their own surface antigens, which is why AB doesn't have anti-A or anti-B. Everybody see the first and third diamonds?
Yes.
Okay. So that explains this. This is what I finished with last time. I need to move on to the RH blood typing system. This has to do with the D antigen. So fill in the blanks there. If you have the D antigen, you're Rh positive. If you don't have the D antigen, you're Rh negative. Again, the D antigen is on the red blood cell surface. Now obviously, if you look at this table, there are only 8 blood types. A, B, and O, all negative. A, B, and O, all positives. There's only 8, and I'm not going to draw like. Everybody can get the 8 from this, right?
Yes.
But this is normal, like the flu shot. Now these people will never make anti-D because that third diamond still applies to the Rh system. Nobody makes antibodies to their own surface antigens. That still applies. But here, these guys can make it. But they're not born with it, because this is like the flu shot. So no one is born with anti-D in their plasma. No one. So I have to put it in parentheses with a big if. If an Rh negative person is exposed to the D antigen, then they will make anti-D. Everybody got it? You see how this is like the flu shot, the norm of the immune system. You don't have antibodies till you are exposed. Do you see how the ABO is the exception? Do you not see it? Do you not get it? I'm not seeing any no's, so I'm thinking everybody got it. Oh, wait a minute. Does everybody get it?
I'm not really getting it.
Okay. Think the flu shot. Rh is like the flu shot. You don't have antibodies for this flu until you get it.
Right?
You're not born with it, you don't have it.
Got it.
That's the Rh system. ABO is born with this without ever being exposed to type B blood. There's nothing else like it. Well, nothing else you guys need to learn about it. That's like being immune to the flu virus without the flu shot.
Right,
That didn't happen. This is the exception.
right.
Does everybody get it?
Yeah.
Right. Well, let's think of it in terms of this issue, erythroblastosis fetalis. Problems arise when Rh negative women carry and give birth to Rh positive babies. Now, you don't need to know anything else about it, just know about the blood type. In physiology, you have to know all about it, so you might want to listen if you end up taking physiology. You have to know about the primary immune response and the secondary immune response. Primary immune response is slow and weak. Secondary immune response is rapid and intense. The flu shot is a primary immune response. It is slow and wimpy. But with the flu shot, you get what are called memory B lymphocytes for that flu virus. So if someone sneezes in the room with the virus, that little bit of virus will trigger this huge secondary immune response, which is why you don't get sick. Already got it? You have to know something about loading dose. If you get the flu shot, you gotta take the whole vial. There's no half doses of flu shots, you gotta take the whole thing. Because the loading dose requirement for the primary immune response is huge. The loading dose required for secondary immune response is tiny. Guy just has to sneeze in the room. You got it? You have to know about, and we'll cover this later this semester, about the exchange between baby and mother and the placenta. There are no open pipes between mother and child, but there's huge amounts of surface area with these things called chorionic villi. Which means every now and then a blood cell will cross, just from the surface area. But not many, because there are no open pipes. Everybody got it? All right, so Rh negative mother has her first Rh positive baby. During the 9 months, red blood cell crosses here and crosses there. It's not enough for a primary immune response. It's not the whole vial of flu vaccine. Nothing happens until childbirth. And I don't know if you've ever seen or been involved in childbirth, but it is at the same time the most beautiful and the most disgusting of human experiences.
Yeah.
I mean, there's fluids going all over the place. Those people got to wipe down and mop down after childbirth because it just splashes everywhere.
Oh my God.
Sometimes babies are born with a brown stinky twin.
Excuse me?
I'll let you guys figure it out.
Oh, yeah.
But there's definitely enough mixing at childbirth for a loading dose for a primary immune response. Everybody got it? Now the mom is now activated. She will now develop memory B cells to make anti-D.
From the baby?
Yes, from the childbirth of the first baby. Not during pregnancy, there wasn't enough of a loading dose.
Can it work the other way around, like if the mom had it, could she pass it to the baby? You know what I'm saying?
If the mom is Rh positive and the baby couldn't be Rh negative, because the Rh negative is autosomal recessive.
Okay.
Anyway, let's go back to this. Let me just finish this story because I got a lot to cover and you don't need to know this for this class anyway. Yeah, she made anti-D, but it was slow and wimpy in the primary immune response. And the first baby don't care anyway. Cords cut, disconnected, baby's fine. Doesn't care whether she has memory B cells. Everybody got it? It is subsequent Rh positive pregnancies. Now the few Rh positive blood cells that can cross over the placenta, rarely, but it does, is like the sneeze across the room. And the mom's gonna generate a secondary immune response, rapid and intense, with huge amounts of anti-D that can now, as this figure is showing, cross the placenta and agglutinate fetal blood in utero. That is erythroblastosis fetalis. Everybody got it? So what do you do for it? You give this thing called RhoGAM, which is anti-D made from a cow. They have inserted human genes in the cow, and the cow will actually make human anti-D. They get the blood sample, purify it, distill it out, so you get human anti-D. And at 3 months, 6 months, and especially right at childbirth, you give mom a metered dose of anti-D from another source, RhoGAM. And you do this for every pregnancy. This anti-D from the cow takes out the D antigen before it can generate a primary immune response in the mom. The mom never has memory B cells. So every pregnancy for this Rh negative woman is immunologically like the first, and the first kid didn't have a problem. Yeah, that was like a campfire story, wasn't it? Now, does the flow of that story make sense? You're gonna have to know the details of the story in physiology. Okay? Any questions? All right, let's move on to the heart. We finished talking about the blood. First, talk about the cardiac conduction system. We did not cover this in lab, so we'll cover this in lecture. While the chambers are contracting, it's called systole. While the chambers are not contracting, it's called diastole. Has everyone here had their blood pressure taken? Did you have two numbers?
Yes.
Like 120 over 80?
Yes.
120 is the pressure when the left ventricle is contracting, which is why the 120 is called systolic pressure. The 80 is when the left ventricle is not contracting, and that is called diastolic pressure. Do those names now make sense?
Yeah.
There are 4 chambers we learned about in the heart. The right side of the heart is going to the pulmonary circuit, left side of the heart is going to the systemic circuit. But I will use this animation in physiology because it's one of the best ones I have. When the ventricles start to contract, they force the tricuspid and bicuspid valves closed. Since those are weak, thin-leafed valves, it makes a soft sound. As soon as ventricular systole ends, the semilunar valves slam shut. Those are much stronger valves, so they make a sharper sound. And classically, when people start listening to hearts in the stethoscope, it's described as lub-dub, lub-dub, AV semilunar, AV semilunar, lub-dub, lub-dub. Everybody got it? One of the reasons why I use this animation in physiology is that if you look at the volume of the blood in the ventricles, 80% of it is just passive drainage from the veins. There are only valves exiting the heart into arteries. There are no valves for veins bringing back blood from the heart. They are just wide open. Can you see that? And 80% of the ventricles is passive drainage from the veins. When the atria contract, it's just topping off the last 20%. 80, 20, 80, 20, 80, 20. Everybody see that? This animation shows really well. 80, 20, 80, 20. Look at also at the contraction wave. The contraction wave for the atria is top down because the direction you want to eject the blood is down to the ventricles. Can you see that? But notice the ventricles are apex up because you have the ascending aorta and the pulmonary trunk going up. Everybody got it? You can see it in the animation, can't you?
And this is why the cardiac conduction system is set up this way, so that you can have 80-20, and you can have the 20 go down to the ventricles. Then you're gonna have 100% of the ventricles going up through the ascending aorta and pulmonary trunk. Got it? All right, remember that the heart wall is 3 coats. Epicardium is visceral pericardium. Endocardium is continuous with the endothelial lining of blood vessels. And they're both really, really thin. The vast majority of the thickness of the heart wall is myocardium. I really like this picture when I found it. How thin the epicardium is, how thin the endocardium is. It's like almost all cardiac muscle with a couple of membranes on either side. Everybody see that in that picture? So this drawing shows the pericardial sac. This is not the pericardial sac, or parietal pericardium is not in this photo. Everybody see the note by the way over there? I finally got the anatomy and physiology tutor hired. This vice president of this college wanted to protect the college, so they redid the employment procedures, and it takes forever to get anybody hired here. So that's why it's now week 11, and we finally have an anatomy tutor. This VP doesn't give a shit about you. Okay. Like all other muscle cells and neurons, myocardial cells carry action potentials. However, these action potentials look different. Don't worry about it, not for our lecture exam 4. But whereas neurons and skeletal muscle action potentials look like spikes, it goes up and down like a spike. In the heart cells, it looks like a slanted box. So it is an action potential, just looks different. Okay, there are 2 things that cause the heart to function like a single unit. And we actually covered this a little bit in lecture exam 2. Skeletal muscle is an anatomical unit because all the cells are fused into these huge fibers. So the skeletal muscle fiber is an anatomical syncytium. This was, I think, in the handouts I gave you for lecture exam 2. Cardiac muscles, the cells are unfused. They are individual cells, but they act like they're fused. And because they act like they're fused when they're not, it's called a functional syncytium. And the 2 things that cause it to function like a single unit are on the slide. Intercalated discs, which have the gap junctions, so adjacent cells share a little bit of cytoplasm. And anastomoses, which is cell branching. And that allows, like this action potential to start at the pacemaker, the SA node, and spread smoothly across all the atria going down, because you got anastomoses spreading out. One cell goes to 2, 2 go to 4. Do you see how cell branching allows this thing to spread out smoothly? Even though the cells are not fused, they act like they are. Functional syncytium. So here's the sequence here. Go ahead and fill in the blanks. The cardiac, this is the cardiac conduction system there in blue, by the way. And a lot of textbooks won't number 2, but I do because otherwise my students tend just to forget it. So that's why I put it in the list, even though a lot of textbooks don't. These are also referred to as nodal tissues. And as I draw on number 5, right and left bundle branches are not symmetrical. Left is bigger, how come? Because the left ventricle is bigger. That's why the left bundle branch is bigger. You guys remember that in the sheet parts? That's why I draw like that. SA node is on the roof of the right atrium. Internodal pathways goes down the atria. The AV node is at the base of the interatrial septum. The AV bundle takes it down to the top of the interventricular septum. Right and left bundle branches take it down the interventricular septum. At the apex, they do a U-turn and go up the ventricular sidewalls in conduction myofibrous. Everything is down, 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 down until you get to the apex, and then all of a sudden it's up. And in physiology, I will tie this all to the ECG wave pattern of P, QRS, and T. And that's what's at the bottom of this animation. P, QRS, T. Okay? And it's just an axial potential going through this series of 6. And because it's a slanted box, that's why the ECG wave pattern looks like that. So that's all in the physiology. Okay? Any questions? There's some animations. Doesn't that kind of look like what I wrote on the whiteboard? Except it doesn't show all the internodal pathways that go over to the left side, because they're all there too. As you can see here. All right, let's go to the blood vessels. We covered this material in lab. So I guess you do need just to know it in lecture. Artery, the term artery and vein is an anatomical term, has nothing to do with the color of the blood in it. It has to do with what it looks like. It's an anatomical term. If it's thick, it's withstanding pressure, the blood is leaving the heart. If it's thin, there is no blood pressure, it's returning blood to the heart. Thick vessels are arteries, thin vessels are veins, regardless of the color of the blood. I covered that, right?
Yeah.
And in the case pulmonary circuit, pulmonary arteries are blue, pulmonary veins are red. In the pulmonary circuits, the colors are switched. Each blood vessel, until you get down to capillaries, has 3 coats or layers. Fill the blanks. We have this in lab in the box for the microscopic images. For lab exam 4.
Oh.
Lab exam 4. Not this week. Okay, now, in addition to identifying the 3 layers, you won't have any pictures on Rutgers on board. But you have to keep track of these elastic membranes, which are not in lab. Tunica intima, or interna, is the endothelial lining of the blood vessels, simple squamous. And it's gonna have some connective tissue, elastic fibers. In larger arteries, those elastic fibers will actually form a layer we call the internal elastic membrane. Okay? It's only in bigger arteries though. Tunica intima is continuous with endocardium. You can't tell where one ends and the other begins, it's continuous. Okay? The intima or interna, either. Better known both since they're both highlighted. Tunica media is a smooth muscle layer. This is the layer that in medium-sized arteries, the vasomotor center, or the vital reflex centers, medulla oblongata. Does this ring a bell at all?
Yeah.
Medulla oblongata, vital reflex centers, there's 3 of them. One of them is the vasomotor center. The vasomotor centers controls the middle layer in medium-sized arteries. And that's gonna cause either vasoconstriction or vasodilation. Again, in bigger arteries, outside of this, you have the external elastic membrane. Okay, engage this, people. If you engage it, you will see that the tunica media is sandwiched in larger arteries between the internal elastic membrane and the external elastic membrane. Do you see that in the words? And then outside of that is collagen. What's called tunica externa. Now in parentheses, there's this word adventitia. Everybody pay attention to this. Adventitia, these are not synonyms. These are tunica intima, tunica interna, same thing. These are not synonyms. Adventitia is a generic term for whenever collagen forms a border of an internal structure. Whenever collagen layer separates a structure from other things inside your body, it's called adventitia. Your ureters have adventitia. Your esophagus has adventitia. 9 out of the first 10 inches of your small intestine, duodenum, has adventitia. Everybody got it?
What do you mean by it separates? Like it was
It's,
together?
it's like a border.
Oh, it physically, it doesn't.
It's a physical border.
Got it.
So adventitia is a generic term, not just for the tunica externa, but the tunica externa is a form of adventitia. Tunica externa is the specific adventitia surrounding blood vessels. I can't really explain it any simpler than that. Everybody got it? All right, now in larger blood vessels, like in large arteries, the walls are so thick. But the fortunate thing in systemic arteries is the blood is oxygenated, and the blood in the artery is actually supplying the cells of the artery. But in really large arteries, it's too thick, and the oxygen and nutrients can't get to the outer layers. And so in really large blood vessels, they will actually have their own miniature circulatory system for the outer layers called vasa vasorum. Okay. So this is the wall of the aorta, and the blood is way over here, like right on in here. And there's no way those cells out there are gonna get oxygen and nutrients. And so way out there in the outer regions is there its own small blood vessels. Does this make sense?
Yeah.
All right, the first arteries is mostly, we reserve these terms for the systemic circuit. The first arteries are elastic. So if you see anything with aorta in it, anything with trunk or common in it, it's gonna be an elastic artery. Now again, they have smooth muscle. When they call it elastic artery, it's not a black and white thing. It just means it has more elastic fibers than the other ones. By percentage, it has more elastic fibers. Everybody got it? Make sure you make that distinction. Elastic fibers have smooth muscle. It's just that by percentage, the wall has more elastic fibers. And so when the left ventricle contracts, it expands. And then during diastole, when it's not contracting, it contracts and gets smaller. It's a controlled expansion and contraction of the diameter. What do you call it if it's uncontrolled? It's an aneurysm. Aneurysm is when, in an uncontrolled fashion, when there's ventricular systole, it balloons out. Then blood is not going down the pipe like it should, it's going into expanding this bubble. And then what's tragic, especially if it's on a really major artery, if the wall gets really thin where it bubbles out, it can burst. And that's usually death sentence. Okay? Why do you think the first arteries need to be elastic like this? I'm just gonna tell you. I'm not gonna wait. The left ventricle contraction is violent. The closure of the aortic semilunar valve is violent. And the blood flow right there in the ascending aorta is violent. Everybody got it? And it's violent throughout all the aorta and all the commons and trunks. Okay? Now, when that aortic semilunar valve closes, the blood flow will actually retract a little bit. If these arteries were PVC pipes instead of elastic arteries, the blood flow would look like this. Boom, 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 and would actually recoil in the artery. Everybody got it? But now in every systole, it expands slightly, and diastole, it contracts. And when it does that, the blood flow looks like this. Boom, 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 boom. It's still moving slower, it just slows up on diastole. It's still moving forward. Everybody got it? That's why the first arteries need to be elastic. Then if you have the arteries with internal and external on them, those are muscular arteries. Again, they have elastic fibers, but by percentage, the wall has much more smooth muscle. These are the arteries the vasomotor center and the vital reflex centers are controlling. Muscular arteries. These are the ones. That's when it's underlined to our autonomic nervous system. That's talking about the vasomotor center. One of the vital reflex centers. Draw a little arrow off to the side of this underlined area. Off to the side, vasomotor center. VRC, vital reflex centers, there's 3 of them. One of them is the vasomotor center, it's this underlined thing right here. Then next to this highlighted words right here, draw a line off and say tunica media. It's the tunica media that's doing the vasoconstriction and vasodilation. And by percentage, these arteries have much more tunica media. That's the smooth muscle. Is it not? Smooth muscle is tunica media.
What was the first one? Oh, sorry, I was right.
Underline right here, vasomotor center. Highlighted words right here,
Yeah.
tunica media.
So when you're saying that's the smooth muscle, you're saying that that's the smooth muscle in the muscular, but in the elastic, there's still smooth muscle.
There's still tunica media,
It's still tunica media.
but by percentage, it's more elastic fibers.
This is the sandwich.
Is that what you're
Yes.
saying?
Yeah, that's why they have elastic, internal and external elastic membranes.
So it's still tunica media.
There's still tunica media. This is not black and white. These distinctions are shades of gray. Does everybody understand the words that are coming out of my mouth? Both of these bullets have both smooth muscle and elastic fibers in shades of gray. Elastic arteries have more elastic fibers. They still have smooth muscle, but they have more elastic fibers by percentage. Muscular arteries have more tunica media by percentage. Any questions? So again, if it's got an aorta in the name, common or trunk in the name, it's gonna be elastic. If it has internal and external, chances are it's gonna be muscular. Then when you get to smaller arteries, you eventually get to arterioles, which are the smallest arteries. The tunica media is no longer a complete layer, but individual smooth muscle cells. When there's a single smooth muscle cell going into a capillary, that's called a precapillary sphincter. They can also respond to vasomotor responses and signals, but this is not VMC, not the vasomotor center. This is more local situations. Okay?
And are those local situations only when it deals with the endocrine or sympathetic stimulation? Or is that just also canvas?
There is a whole local conditions that can be beyond hormones and autonomic. But endocrine would be a good illustration of this. Let's see if I got it over there. Yeah. So I'm showing you plans, let me explain this in terms of the endocrine. Everybody got the blanks? Once you get to the capillaries, there's only one coat left, and that's the tunica intima. That's the only one left, and that's the endothelial layer, the basement membrane. The cumulative diameter at the level of the capillaries is huge. Even though each one is tiny, in fact, they're about the same diameter as a red blood cell. Out of your trillions of cells, all of them are close to a capillary. You just have so many capillaries. Are we clear? And so at the ascending aorta, it's like an inch pipe. Once you go through the aortic arch and the three branches, the cumulative diameter is a little bit bigger. Once you get to the head and the arms, it's a little bit bigger. Once you get down into the chest, it's a little bit bigger, and all the way down to the legs. You're getting into smaller arterioles. The cumulative diameter is getting huge. At capillaries, it's the biggest. Everybody got it? Started out here with the ascending aorta. By capillaries, you're like this. Because of that, and you don't need to think like a physician, you just have to think like a plumber. If that happens, the water flow is going to be really slow. Okay? If you got a 1-inch pipe going into a house, unless you have other sources of water, if you spread it out too thin, pretty soon each faucet, if it's running, it's not going to give you very much flow. Everybody got it? Well, that's good you want it slow there, because diffusion takes time. And so that's advantageous for it to be so slow with capillaries. Okay? Now let's look at this capillary bed. There's this thing referred to as the meta-arterial, or thoroughfare channel, or preferred channel. Those are all synonyms for the same thing. It is the capillary with least fluid flow resistance. The meta-arterial thoroughfare has the least fluid flow resistance. All right? And this is part of that micro-control of circulation with the precapillary sphincters. Let's say, for example, we just gone through this lab exam. Well, Thursday you still got it. But in the endocrine, is it pancreas or pancreatic islets? Pancreatic islets. And they're tiny. And let's say right here is a pancreatic islet. When you are not eating, and you don't have a lot of blood glucose going into your blood, all these other precapillary sphincters to these smaller capillaries are constricted a little bit. And the one through the meta-arterial is dilated. So the majority of the blood is just going through the meta-arterial, straight to the venule. You see how it is a thoroughfare channel that, boom, straight to the venule. Everybody see that? Yes. That flow is enough to keep the pancreatic islet alive. So you just have it go through the meta-arterial. So far so good? Now you eat a Snickers bar and your blood glucose spikes. Your beta cells in your pancreatic islets wants to secrete insulin. But there's a bunch of cells making insulin. So if this capillary bed needs to pick up a bunch of insulin, it's going to constrict the thoroughfare channel, the meta-arterial, and it's going to dilate all the ones to the rest of the other capillaries. Now you have a much slower blood flow through this pancreatic islet to pick up insulin. Does that answer your question? Everybody get the micro-control of blood flow through a capillary bed and the precapillary sphincters. Everybody got it? Again, most capillaries are continuous. There are continuous capillaries, capillaries with pores called fenestrated. And a good example of a capillary that would be fenestrated would be that example I gave you in a pancreatic islet. Insulin is a quaternary protein, it's big, and it'll have difficulty getting through unless you've got pores. Everybody got why you need fenestrations? Okay? There's a third kind of capillary, and that has huge wide gaps. Just barely able to keep blood cells inside of it. And that would be a sinusoid. We are going to see liver sinusoids next week. Yeah, next week in lab, not this week. Tuesday lab, you remember the hepatic portal vein? You guys remember that? Two capillary beds. It's a capillary bed in the stomach, and then it goes through the left gastric vein into the hepatic portal vein, and then it goes through a second capillary bed in the liver. Does this ring a bell? Portal vein is between two capillary beds. Everybody look at me. Everybody look at me. On the hepatic portal system and the hepatic portal vein, the liver sinusoids are the second capillary bed. These are types of capillaries. There's three types. Okay? So, oh yeah, Tuesday lab, I said, challenge me and give me a point A in the body. I'll take it, and I'll take it to a point B, and I'll go through the vessels on page 30, and I don't have it memorized. Somebody give me point A. Come on, anywhere in the body, come on. Make it fun.
Elbow,
All right, which one?
your left elbow,
Left elbow, okay, that's point A. How about point B?
your right ankle.
Right ankle. Left elbow to right ankle. That's too easy.
Okay.
Left elbow to right ankle through the liver. All right, here we go. Left elbow. Let's say if we're on the medial side of the elbow, we're going to go into the ulnar vein. You're going to go to the brachial vein. Going to go to the left axillary vein, left subclavian vein, left brachiocephalic vein, into the right atrium. Right atrium, tricuspid valve, right ventricle, pulmonary semilunar valve, pulmonary trunk. Let's go to the right lung. Right pulmonary artery gets oxygenated, right pulmonary vein, left atrium. Bicuspid valve, left ventricle, aortic semilunar valve, ascending aorta, aortic arch, descending thoracic aorta. I gotta go through the liver, right? Descending abdominal aorta, celiac trunk, common hepatic artery. I'm gonna go through the liver sinusoids. Say I'm in the right side of the liver because it's bigger. Right hepatic vein, inferior vena cava. Right atrium, tricuspid valve, right ventricle, pulmonary semilunar valve, pulmonary trunk. Let's go to left pulmonary artery, oxygenated, left pulmonary vein, left atrium, bicuspid valve, left ventricle, aortic semilunar valve. Ascending aorta, aortic arch, descending thoracic aorta, descending abdominal aorta, right common iliac artery, right external iliac artery, right femoral artery, right popliteal artery, right posterior tibial artery to the ankle. I don't have page 30 memorized. This is what I'm trying, this is the reason why I teach arteries going out from the heart, veins coming back from the heart. If you think it through sequentially, you don't have to memorize page 30. I don't have it memorized. It's kind of like driving here to Disneyland. Who here wants to go to Disneyland?
Sure.
You could probably name the freeways it's going to take to get there. You don't have it memorized, but in your head, you're just imagining the drive. Imagine yourself a red blood cell in these pipes and go to Disneyland. My Disneyland was the right ankle. Does that make sense?
Yeah.
Well, anyway, the liver capillaries have huge gaps, those are sinusoid capillaries. Huge gaps, so continuous, tight junctions, fenestrated's got pores like in the pancreatic islets, and then in various places, spleen, liver, bone marrow, you have huge gaps. Those are sinusoids. Now this is showing you how things can get in and out of the capillary. There are three, make sure you pay attention to this. On the left diamond, there are three ways to do diffusion. You can either diffuse straight through the endothelial cell. You gotta be able to cross the lipid bilayer though. Not everywhere do you have tight junctions. Now in the brain capillaries, they're pretty much everywhere, but outside the brain, you can have some spots that don't have tight junctions. There will be intracellular clefts. It can diffuse through those. Or if it's a fenestrated capillary, it can go through a pore. So three subforms of the left diamond. It takes energy, but especially if there's a substance that's got to go through and it can't get through the lipid bilayer, it goes through endocytosis on one side of the endothelial cell, exocytosis on the other side. And it can get through that way. That requires ATP. The three options of the left diamond do not. But the right diamond requires cellular energy, ATP. Okay? Venules are the smallest veins. We will see the return of tunica externa. And in veins, the percentage of the wall that's tunica externa is actually quite a lot. Small veins will increase in diameter. Again, you'll have more tunica externa, less smooth muscle. Tunica interna is always the same, so even in arteries. Now it's really thin walled because there's no blood pressure. Once you go through this huge cumulative diameter in capillaries, the blood pressure drops off and it never recovers. Okay? And that's why you gotta have valves. If you are standing, there's not enough hydrostatic pressure in the veins to return blood to your heart. Okay? The only way blood can go up against gravity is through valves. And to have valves work, you have to move around. You have to contract skeletal muscle. And when you contract skeletal muscle, one side closes, the other side opens. And that's how it can go up against gravity.
Sorry.
What is
It's
it?
not, it's a smoothie I need. No, it won't come out.
As you see when I'm trying to get the last bit of whiskey out of my body. Then all the ice just hits me in the face. Okay? Now, if the valves start to fail, then that's where you get varicose veins. Any questions? Yes?
You said that you have to move in order?
Uh-huh. Well, can I give you a good example of this? All right, I coach kids basketball and Paulding Middle School basketball for Robin Ventura. He had daughters, and I coached 3 daughters for them. Well, we had a whole bunch of guys going to see him play while he was still playing for the Yankees. And we took a red eye to New York, and I'm 6'2", and if you're 6'2" and coach, man, you're not moving. But I just wanted to sleep because it was a red eye, so I said, I'm gonna take off my shoes. Stupid mistake if you base it off of the slide. Because what is the one plumbing principle all plumbers have to know?
Things go down, flow downstream.
Shit rolls downhill. You guys gotta know that, man, because that'll be big in physiology. If you can remember plumbing principle, shit rolls downhill. Basically means the blood is gonna go to the lowest part of my body, which were my feet. While I'm sleeping, every beat, my feet are getting bigger. I can't contract muscles to return it to my heart. Finally, the guy goes, dude, we're landing in LaGuardia. Time to get up. So, oh, right. I tried to put on my shoes and I could not get my shoes on. So I had to walk the baggage claim
Oh my God.
with my heel outside my shoes. And back then, this was before, all the security stuff with 9/11. And this guy, there was like 19 of us wearing Yankees hats. And this guy kept asking me for an autograph. And I'm going, dude, we're not the Yankees, all right? We're not gonna fly coach, all right? Well, I'm walking down to the baggage claim. Because of the walking, I was contracting skeletal muscle. My feet, even though they were the lowest part of my body, were shrinking with every step. So by the time I got to baggage claim, I could put my shoes on. Okay? And this guy asked me for an autograph again. I'm going, gee, we're friends of one of the players, we're not on the team. And I hardly got any sleep. I should have just signed this baseball. My boss loves you. Can you just sign this baseball? I should have just signed it with my name. And after the first game, I told Robin what happened. He started laughing because we both figured it out. There are 8 million people in New York. There were only 2 6'2" Japanese guys, me and Hideki Matsui. And he goes, Matsui is the butt ugliest guy in the major
Oh,
leagues.
no.
He's got a size 8 helmet. He has to have his own batting helmet because it's size 8. So he gets up to bat in the next game, and his picture comes up on the Jumbotron, and I'm like, damn, I was insulted. There's some varicose veins. Okay, circulating routes. We talked about the 2 pumps, double pump. Right side of the heart pumps to the pulmonary circuit. Here, the colors are reversed. Pulmonary arteries are blue, pulmonary veins are red. This circuit is dominated by capillaries because it's, it's very short. It's a very short loop. Hardly any pipes, but as soon as you get into the lungs, boom, it's all capillaries. So it actually holds quite a bit of blood. It's just going really slow, which is good because you want to get rid of CO2 and you want to pick up oxygen. That diffusion takes time. So you want the blood flow to be really slow in the pulmonary circuit. Systemic circuit starts with the ascending aorta. There's really one way out of the heart in the systemic circuit, and that's ascending aorta. There are 3 ways back. What are the 3 ways?
Diffusion? There's 3 ways of diffusion.
3 veins. 1 artery out, ascending aorta, 3 veins back.
Oh my God. I don't know.
Well, 2 of them are up there.
Superior vena cava.
1,
Inferior vena cava.
2,
Extending aorta.
no, that's 1 out.
Okay. Cells.
Think-
Atrium, right atrium? Oh, that's.
Think right atrium. That's where the systemic circuit returns. Right atrium.
Is this the right atrium?
Superior vena cava, inferior vena cava, and
Coronary artery.
okay, I don't want your brains to bleed. Coronary sinus.
Oh.
Those are the 3 veins that drain into the right atrium. Superior vena cava, inferior vena cava, coronary sinus. Ooh, I was like pulling teeth. And that's this here. So the coronary arteries are not counted as ways out because they come out of the ascending aorta. So ascending aorta is the only way out into the systemic circuit. But all these capillaries, then venules and veins will all collect in the coronary sinus. So here's the 3 right here. 1, 2, 3. Everybody got it?
Can you point those out one more time?
Superior vena cava, inferior vena cava, coronary sinus, all going into the right atrium. underlying there is the reason why coronary arteries tend to get more atherosclerotic plaques than other arteries. You can get plaques anywhere, but you tend to have greater buildup in places of the highest pressure and most turbulent blood flow. That's why it's underlying. That has clinical relevance. And then here's the hepatic portal. Just gonna go and fill in the blanks. This drawing's not really good though, because it doesn't look like a chair. Remember how the hepatic portal system looks like a chair?
Yes, they do. Oh, they didn't get that yet.
Oh, yeah, Thursday lab, huh?
Yeah, yeah.
Oh, that's today.
Okay, I was like, damn, I'm not remembering anything.
And again, this, and what I said in lab on Tuesday is that your body is designed so that all the blood from the stomach to the rectum will filter through the liver before going into general circulation. Someone from Tuesday lab, can you tell me why? Because people eat and drink stupid shit.
Oh, yeah, right, you know that.
And the liver filters out a lot of the bad stuff before it goes into general circulation. That's why.
The liver, love your liver. Liver does a lot.
Yeah. But I'm gonna abuse my liver. Okay, any questions? So, here's the two capillary beds. Remember I said a portal system is between two capillary beds? The first capillary bed is like, say, the stomach, or the large intestine, or the small intestine. That's the first capillary bed. It's gonna go through the hepatic portal vein to the liver sinusoids. The liver sinusoids are the second capillary bed. Are you connecting these?
Yeah.
Let me give you one other piece of advice for lecture exam 4. The more you engage to understand, the more this is going to be doable on finals week. It is a lot of stuff. If you think you can be disengaged and then all of a sudden, just before lecture exam 4, you can put it together, you won't. Engage now, make it make sense now. And even if you forget it, when you are studying for lecture exam 4, and this is true for all big exams, it'll come back to you much faster if you make it understandable now. Oh, I remember that, because I understood it. If you are disengaged, then you look back 2-3 weeks before, it's gone. You have to have a thought process to get this information encoded in your brain. Make it make sense. Okay? Any questions?
Will that last one make a little more sense on Thursday instead of like there?
Which one? Oh, the hepatic portal system?
Yeah.
Yes. Yeah, on Thursday lab, people, if you have textbooks, bring them to lab, okay? Because we'll be looking at things in the textbook. How many textbooks did we have in Tuesday lab?
Well, I had one, but I just left it in my car, remember?
Oh, yeah.
I was the only one that did that.